Hello champ. So today it's our third session and today we'll also practice 10 question in 10 minutes. Okay. So without wasting a single second, okay, let's jump into the question. Our question number 1. The signal for ER resident protein contain PK3RKV at N terminus of the protein, KDEL at N terminus of the protein, KDEL at C terminus, SKL at C terminus. So I hope you know that suppose this is a protein and the protein contains some signal peptide, maybe at the N terminal or the C terminal, that actually determine that the destination of the protein molecule. Now the question for ER resident protein, what is the particular signal for this ca- uh, sorry, for this ER resident protein? Okay, now we'll try and start. 1 2 3 4 and 5. Okay, the answer is KDEL at C terminus. Okay, the PK3RKV at N terminus, that's general signal peptide for nucleus, and SKL at C terminus, that's for peroxisome. Okay, now our question number 2. Which of the following lipid is flipped during apoptosis? First option is phosphatidylserine, second option is phosphatidyl inositol, third option is phosphatidylethylamine, fourth one is sphingomyelin. Okay, try, pause and try. 1 2 3 4 5. So the correct option is, yeah, of course, the PS, the phosphatidylserine. So suppose it is cytosolic part and this is the extra cellular portion. Okay, so during apoptosis the PS is flipped from inside to outside. Okay, so PS and SM, PE and PI, right? I hope you know that. Okay, sorry. Now gated transportation of protein is observed in case of cytosol to Golgi, ER to Golgi, cytosol to lysosome, and cytosol to nucleus. There are three different kind of transportation. One is gated, another is through vesicular transportation, and another through transmembrane transportation. Right? So which one of the following is gated transportation? Your time start. 1 2 3 4 5. The correct option is from okay, so from cytosol to nucleus, that's the correct option. There is nuclear pore that will control the gated transportation. So fourth question. So the fourth question is the cytochrome oxidase is marker enzyme of which of the following cell organelle? Endoplasmic reticulum, peroxisome, nucleus, mitochondria. Okay, this is very easy question. Okay, 1 2 3 4 5, that is mitochondria. So for ER, that is PDI is the marker enzyme, protein disulfide isomerase. For peroxisome, you can say any kind of SOD, superoxide dismutase. For nucleus, you can say histone. Right. Now question 5. Transportation of ER resident protein from ER to Golgi is mediated by COP1, COP2, glycine, caveolin. I hope you remember. So suppose this is ER and then it's Golgi, right? And from here, suppose this is plasma membrane. There is some default pathway, okay, and this is lysosome, right? Now recall and say what's the exact thing. Okay, this is COP1, COP2, glycerin and caveolin. Okay, you can comment, right? I can tell you a different thing, glycerin, caveolin, anything is possible. Just think and comment. We will leave some question for your comment. Now question number 6. In which phase of mitosis ER and Golgi is fragmented? Prophase, metaphase, telophase, anaphase. Okay. Uh, I the all the phase, there are some different changes in cell cycle, right? So the something happened in prophase, something in anaphase, something telophase and anaphase, uh, metaphase. So the question is, in which phase of mitosis ER and Golgi is fragmented? The correct option is 1, 2, 3, 4, 5. Yeah, prophase. Now question number 7. Which of the following DNA polymerase have 5' to 3' exonuclease activity? Should I leave it for comment or okay? So option A DNA pol 1, option B DNA pol 2, option C DNA pol 3, option D all of the following have 5' to 3' exonuclease activity. Just think and try to imagine what is the function of 5' to 3' exonuclease activity. Okay, the correct option is 1, 2, 3, 4, 5 DNA pol 1. The 5' to 3' exonuclease activity is generally responsible for primer removal. Ideally, I should leave that type of question, right? Now, question number 8. Which of the following mutagen cause tautomerization? So, ketoenol tautomerization, right? Keto. So, suppose from keto to enol, that kind of things. That is called tautomerization. So, this is ethyl methyl sulfate. This is acrid in orange. This is HNO2. That is fibromeuracil. Your time start now. 1, 2, 3, 4, 5. So the correct option is HNO2. That actually cause that is deaminating agent that will remove the amine group, and through that it will cause tautomerization. Ethyl methyl sulfate that actually responsible for methylation. Yes, acrid in orange. That is all uh, intercalating agent. Fibromeuracil that is the base analog. Question number 9. 5S RNA is synthesized by which of the RNA polymerase? RNA pol 1, RNA pol 2, RNA pol 3, or none? Uh, you should comment, I think. Okay, uh, let's try. So first, quick. So the 5S RNA is synthesized by which of the following RNA polymerase? Pol 1, pol 2, pol 3, none. The correct option is pol 3. So pol 3 transcribe tRNA and 5S rRNA. The rest of the RNA, suppose 18S, 5.8S, 28S, all are synthesized by RNA pol 1. Okay, and RNA pol 2 synthesized mRNA and siRNA. This is very important thing. And which of the following antibiotic block peptidyl transferase activity on streptomycin, tetracycline, chloramphenicol, puromycin? Which of the following antibiotic block peptidyl transferase activity? Streptomycin, tetracycline, chloramphenicol, and puromycin. 1, 2, 3, 4, 5. Chloramphenicol is the correct answer. It stops the peptidyl transferase. Peptidyl transferase means, suppose in case of what happened, this is ribosome, this is suppose the largest subunit, this is smallest subunit, there is some tunnel. Okay, now one peptide is one amino acid is coming and it's attaching to the uh, growing chain, right? So the chloramphenicol will stop it. Okay. Okay, so our 10 question is done, and you have to calculate honestly out of 10 question what is your score. If it's above 6, your preparation is quite good. Okay, we will meet tomorrow with another 10 question.